Sorry about that, guys. All right, well, welcome to week 8's review. We'll be going over the regulatory pathways in S, G2, and M phase. All right. So a quick overview of the entire cell cycle is given here on the slide. Yeah, so first we started off with G1 over here. Can you guys see my mouse right here? Yeah, so I guess, yeah. So G1 is basically what we were talking about last week. And I guess like G0 going in out of like G0 into G1. And we kind of like briefly just like skimmed over the rest of it, but now we're going to start talking about the regulatory pathways within each one. So S phase, now that we've moved beyond the restriction point, that was at the end of G1, and the cell has now started to move into S phase. Once the pre-RC is assembled, the other proteins are added to the pre-IC complex. And so this has been formed, and then once the origin of replication fires and the DNA replication begins, S phase is entered by the cell. And as noted down here, not all origins will fire at the same time. Some will fire earlier than others, but when the first ones start to fire, that's when the cell has entered S phase. And just as a clarification, I got some questions on this. There isn't one origin of replication that is on each chromosome. There's like many along the chromosomes that come together to like meet each other. Okay, so the first step in S phase like regulation is that SCDKs will inhibit new pre-RCs from forming. And this is because you only would want the DNA to replicate once per cell cycle. You wouldn't want to make like two of like chromosome like 22 or however many chromosomes there are. You don't want to make like three, or sorry, you don't want to make like three of them, you know, more than two will not be good. You get like an extra chromosome. And so S cyclin / CDKs do three things to prevent the formation of new pre-RCs or to prevent the refiring. And so the first one is the phosphorylation of ORC to prevent the binding of CDC6. The second one is the phosphorylation of CDC6 itself and CDC6, which this leads to the degradation of CDC6 through ubiquitin-mediated proteolysis, so that the MCM complex cannot be recruited. And the third thing is that it will phosphorylate MCM27, and this causes it to be exported for nuclear export. Exported from the nucleus. And so I definitely would keep this in mind right here of the phosphorylation of MCM and what happens to it, because I remember from previous like years, like this is definitely like a test question that has thrown people off, where you know you're focused on all these other things, but it's definitely good to make sure you've got these details down of what happens to MCM. And keep in mind, like all of this, the reason why it's exported from the nucleus is because all the stuff that we're talking about right now in the cell cycle isn't taking place outside of the cell when it comes to like DNA replication. Like all this is, it's not happening like in like the general like cytoplasm area. It's all happening in the nucleus itself. And so that's why being exported from the nucleus would prevent it from allowing pre-RCs from forming. And so the first thing that's going to be happening another thing that's going to be happening in S phase is that the cohesion rings are going to form around the replicating DNA. And this keeps the strands together throughout and together like throughout the process of S phase and through G2 and into M phase until they line up at the mitotic or in between metaphase and anaphase. And so this prevents the chromosome segregation during the spindle assembly, and it ensures that an equal number of chromosomes are on each side for division. And the cohesion rings must be present in S phase in order to function in M phase to help them line up. Oops. And at the end of S phase, there's a checkpoint that checks for if there's DNA damage. It also checks for if the DNA is completely replicated. We can't just have like some of the chromosomes are replicated and then some of them still only have one copy. You have to make sure that the entire genome has been replicated. And then P53 activation can also happen in this phase as well, just like we talked about in G1. If there is DNA damage, P53 can be activated to either fix that or cause cell death. And so replication can be stopped by this checkpoint temporarily if necessary to like help, like I said, like fix the damage if there's any damage done, or if there is like some pieces that still need to be replicated. Now moving into G2 phase. So once the S phase checkpoint has been passed, the cell has duplicated its entire genome. And now the cell is focused on growing again and preparing for chromosome segregation, making sure the DNA is done being replicated and it's already been checked for DNA damage. Oh yeah, so during this step, it's also checking as well for DNA damage and like I was saying before, like an S phase at the checkpoint, make sure it's completely replicated. And this growth is really important because it's like all the growth it's going to get before the cell divides. The cell needs to make sure that it's got like double of everything, double like the cytoplasmic space or the organelles and all that stuff. And if there's no activation of M phase CDK, there is no entry into M phase. And so the transition into M phase is dictated through this G2 checkpoint. And this is definitely a pathway like I would recommend having written out or having drawn out on your cheat sheet for the exam. So MCDK levels are rising throughout this G2, but they are inactive in like at the beginning of G2. So at first there is like MCDK like cyclin complex down here. There can be a lot of the levels can be high in the cell, but it's not going to move. The cell isn't going to move into M phase because it's not phosphorylated. And even if it is phosphorylated by CAK or activating kinase, it's also going to be going to have an inhibitory phosphate placed on it. And so those together will make MCDK inactive. So here just talks about that it's being held inactive by WEE1, like I just said. And yeah, we need to have the cells grow big enough. So we don't want to have MCDK cyclin active just quite yet to move it to M phase. So later in the G2 phase, again, I'd probably also have this written down, especially this right over here. After the checkpoint, the cell activates MCDKs to proceed to the M phase. And so this process is started by CDC5 being partially activated by another kinase. And so this phosphatase is actually an activating phosphatase because a lot of times we talk about in this course, like the phosphatases are like removing an activating phosphate, but in this case, and so it usually deactivates things, but in this case it's different because it's removing an inhibitory phosphate from the MCDK. And so by removing this inhibiting phosphate that was placed on there by WEE1 previously, MCDK is now active. And so the reason why this causes like such a huge spike in MCDK or M-cyclin CDK activity is because M-cyclin CDK, when it itself is active, causes this feedback loop where it will activate, it'll fully activate CDC25, and which causes the CDC25 phosphatase to activate even more inactive MCDKs, which then causes, and as there are more active MCDKs, those continue to help activate these phosphatases that help activate themselves. And so this causes like an exponential increase in M-cyclin CDK activity because it's causing something, or has like a downstream effect that like helps it upstream. And the other way that this positive feedback loop is like further enhanced is the fact that active M-cyclin CDK will also phosphorylate and activate WEE1. So there will be less WEE1s placing that inactivating or that inhibitory phosphate onto MCDKs, and so there will be less inactivation of it. Thus, will be a lot more active MCDKs around. And so that, like I said, further accelerates this positive feedback loop. Okay, and so now that there is high levels of cyclin CDK activity, the cell can move into M phase. And so here's just a quick overview of M phase in its totality. So there's the beginning, there's prophase where active MCDK causes the breakdown of nuclear envelope and chromosomes condense. Then in prometaphase, between prophase and metaphase, where the microtubules will grow and shrink towards their respective locations and look for proper kinetochore attachments. And then at metaphase, chromosomes will align along the metaphase plate here, and the chromosomes will have proper attachments. And then, of course, there's the spindle assembly checkpoint, which occurs between the two, metaphase and anaphase. And once it passes this checkpoint, it can go from metaphase into anaphase, where the chromosomes are pulled to the spindle poles, and the separation of the duplicated chromosome occurs. And then finally, telophase and cytokinesis are when the nuclear envelopes start to reform, everything starts to go back to normal, the chromosomes decondense, and the cell is like dividing up here. And then there's two cells. And then after this, of course, each one of those two cells will go into G0 or G1. Okay, but now we're going to go into each one of those phases in much more detail. Active CDK. So active, whoops, sorry. Oh my God. Okay, so active MCDKs, the way that they cause the transition into prophase, which is the first step of M phase, is a few ways. So the active MCDKs will cause the lamins to, they'll phosphorylate lamins to break down the nuclear lambda. So basically just like breaking down like the nuclear envelope. And then it'll also cause phosphorylation of condensin to induce chromosome condensation. It'll make them look like going from spaghetti that's just like loose and everywhere to sausages to be so that they can be lined up at the metaphase plate and pulled apart. And you can't have the nuclear envelope there, and the reason why it's like broken down is because like you can't pull the chromosomes to like two different like sides of the cell and then like you can't pull them to two sides of the cell if there is a nucleus in the way. So that's why it has to be like broken down. And the active MCDK will also phosphorylate various maps and motors to facilitate the mitotic symbol assembly. So the idea with sequential phosphorylation, which is what's occurring basically to push the cell, part of like that process back here of pushing the cell into M phase, is that phosphorylation needs to happen in a certain order. So CDKs are a master like cell cycle regulator. They act as like a priming kinase for downstream kinases. And so inhibiting CDK will prevent the cascade and halt the cell cycle any phase is needed, which is nice because all you have to do is like inhibit one type of kinase, and then the rest of the cell cycle can be paused. So it's like the master like coordinator, regulator. And so on the left here, you can see substrates of PLK1. They need to be phosphorylated by CDK in order for PLK1 to bind to the substrate. And so what this is showing and like how this connects to the, how this connects to the previous part of like moving into M phase is like, it helps like phosphorylate other, like we are showing here, like it helps phosphorylate other proteins that help carry out their own functions in order to like help out with whatever is needed. So like here, there's like several things that need to be done in order to have the cell move into M phase. And so while CDK might not be doing individually all those things, it's the master regulator in terms of like helping signal to all these other proteins to help like cause these like multiple downstream effects and simultaneously. And so the spindle assembly comes up next. So there is microtubules will assemble into spindles, and the chromosomes need to separate in the beginning of M phase. So right here, each one of them needs to be attached at the kinetochore, which is the center of the chromosomes once they've condensed. And so along here, they're lined up so that each one, and so, yeah, they're lined up so that there is one chromosome of each, well, there's two of each, but one pair at each spot here lined up. And so these chromosomes right here are all condensed, and they all still have the cohesion holding them together until the spindle assembly checkpoint is passed. And so they're all lined up by having these microtubules attached to them at the kinetochore. They're the center of the chromosomes. And then there's also other chromosomes that are used, or sorry, other microtubules used to help hold this whole spindle assembly in place. So right here you have the spindle poles, which is where the all the microtubules are connected to, at least on one side. And so the first ones I was talking about were the kinetochore microtubules, which will later be used to pull the each chromosome to like their respective side. And so these will form like their own cells on each side. Then you have the aster microtubules, which grow from the spindle pole and attach to the cell cortex. the kinetic core ones, like I said, they grow from the spindle pole and attach to the kinetic cores of the sister chromatids right here. And the interpolar ones are the ones that are just they're not connected to the sister chromatids themselves. They are overlapping in the middle and play into the push-pull mechanism that we'll talk about soon. And so all of these, keep in mind, like they grow from the spindle pole, and so their negative ends are going to be in here. And the positive ones will be growing outwards. And so the kinetic core is a large multi-protein complex built on the centromere of each sister chromatid. And so the centromere is a region of the chromosome right here as shown. And it consists, the centromere consists, or the kinetic core consists of 80 different proteins. And the spindle microtubules will also be attaching to specific proteins within the kinetic cores themselves. And so here you can see like a zoomed in version of like these sister chromatids lined up. You've got each side has a chromatid, like a copy of a chromosome. And they'll all be like lined up right here. And they'll be pulled apart after, of course, like the spindle assembly checkpoint. But for right now, they still have the cohesion wrapped around them and they're still tied together. Okay, now for this push-pull theory or mechanism, there is opposing forces from the motor proteins that cause the chromosomes to line up. And so the spindle formation requires an array of opposing forces from kinesins and dyneins. And so the kinesins are the plus end directed motors. So remember, they're walking towards the plus end, sorry, of the microtubules, whereas the dyneins are walking towards the minus end. And so those are walking towards opposite ends of microtubule, and they cause opposing pushing forces that stabilize the spindle. An example of this is EG5, which is the motor protein that binds the interpolar microtubules that overlap. They're plus end directed, and they push the spindle poles closer together. So as you can see here, each one of these, so EG5 is unique in that it connects to or walks along two microtubules at once, rather than having one end that is attached to a vesicle and just in like another that's attached to the microtubule. And so these will be like pushing the spindle poles closer together, as you can see here. As they walk towards the plus end, they will be pushing the poles. Okay. And so the sister chromatids are at this point, when they're in metaphase, which is when they're lined up along in the middle of the cell, they are still secured by the cohesion rings. They're attached to the microtubules at the kinetic cores. And they have equal and opposing pulling forces, and that causes them to line up along the metaphase plate. And so the transition from metaphase to anaphase requires passing through the spindle assembly checkpoint. And so the checkpoint ensures that all sister chromatids are aligned and the spindles are attached properly at the kinetic core. The cell will inhibit anaphase unless there is proper microtubule attachments at the kinetic core. But otherwise, non-disjunction would occur. So the main idea about this spindle assembly checkpoint is that the MAD and BUB sense microtubule tension on the kinetic core. And when they sense that the tension is equal, that's how they know that all the chromosomes have lined up in the middle of the cell, and then that causes them to release CDC20, which should not be confused with CDC25, which was the activating phosphatase at the end of G2. So CDC20 is released. Once it's released from MAD and BUB, this causes, or this activates, binds to and activates APC, which is a ubiquitin ligase. And so this ubiquitin ligase will target the protein securin, and which is right up here, which has been holding separase almost like hostage. And so it'll grab onto and target securin for ubiquitination. And once it's ubiquitinated and degraded, securin, that allows separase to be free and active. Once it is active, it can now cleave cohesion, which the cohesion rings and the sister chromatids can separate, and that causes anaphase because there already was tension on the kinetic cores. The only thing holding them together here at the metaphase plate, the chromosomes or the sister chromatids together, was the cohesion rings that are shown here or in more detail here. So securin is breaking these cohesion rings. Separase is causing the degradation of, or cleaving the cohesion rings, and the sister chromatids are now able to separate. And this is the transition between metaphase and anaphase that is named Spindle Assembly Checkpoint. APC also targets M-cyclin for degradation, and this causes a decrease in M-cyclin CDK activity. So that this is pretty key because it'll cause, once M-cyclin CDK activity drops, then it'll stop phosphorylating the lamins and condensins that cause the transition into M phase, so it can start going back, making the transition out of M phase once anaphase and the rest of the phases of M phase are done. And so there's two parts of anaphase. There's anaphase A and anaphase B. So anaphase A is when the chromosome is a part of anaphase where the chromosomes are pulled towards the poles. And so the shortening of the microtubules causes, or these forces are generated at the kinetic cores by the motor proteins, and this causes the movement of the chromosomes towards the spindle poles as they're walking along the microtubules. So chromosomes are pulled poleward. Yeah, they're walked by these minus N directors, because remember the minus N director, or the minus N is at the spindle pole, because the microtubules grow from their minus end here at the spindle pole out to the positive end. And so these are walking towards the minus end, and as they walk, these microtubules shorten, and the spindle poles stay in place, but these are being pulled towards them, towards the side. And the anaphase B portion is referring to the rest of the forces in anaphase, causing and this focus is more on the forces on the poles themselves. So in anaphase B is focusing more on the poles being pushed and pulled apart. I believe earlier in the presentation it said that these overlapping ones was causing the poles to oh wait, no, it did. Yeah, it was causing them to come together, but I believe it is causing them to be pushed apart here. And so both here, the overlapping ones and the motor proteins here and the motor proteins but here are causing forces that cause the spindle poles to be pushed and pulled apart. And so the sliding force is generated between the interpolar microtubules from the opposite poles and push the poles apart. And then there's a pulling force, pulling from up here, and it's acting directly on the poles to pull them apart. And the microtubules are actually growing here at the plus ends of the interpolar microtubules in order to make sure that the cells are able to separate and become like their own distinct cells. And so the spindles lengthen, they grow on the plus end. Yep, the poles are pushed and pulled apart. There's a sliding force between the interpolar microtubules, and yeah, the pulling force directly pulls on the poles to pull them apart from the outside here. And so now we're starting to move into telophase and cytokinesis. Once the sister chromatids have reached the opposite poles on either side, a cleavage furrow is formed. And so the way that it happens is it's created by this contractile ring in the middle made of actin and myosin. And it causes an indentation in the cell's membrane that gradually deepens until it hits the spindle, and that is left between the two nuclei, and eventually the splits, and causes there to be two distinct cells. And so it's important to remember that the M cyclin CDK activity had dropped earlier, like I mentioned, and so because of that, the lamins and the condensins are no longer causing, the breakdown of the nuclear lamina and the condensing of the chromosomes. And so what happens is that the spindle begins to disassemble, the chromosomes start to decondense again, going from like the sausages to now back to loose spaghetti, and the nuclear envelope is able to reform around the DNA and reforming the nucleus basically. And so the main ideas from the cell cycle from today are that it happens in a strict sequential order: G1 and then S, then G2, then M, and then it goes back to G1. And of course, this M phase right here is broken up further into prophase, prometaphase, metaphase, anaphase, and cytokinesis, and telophase. And so that was the last step there. And so the cell cycle events often they involve cyclical activation, deactivation of proteins, and often by means of phosphorylation with ATP. And a lot of times this is done by CDK and other proteins that are activating, deactivating CDK. CDK must be bound to cyclin and phosphorylated by an activating phosphate, but not an inactivating phosphate, in order to be active. A CDK regulates various proteins in different phases of the cell cycle. So in different phases of the cell cycle, they have their own cyclins, and each cyclin is created and degraded in each part of the cell cycle in order to control what part of the cell cycle the cell will go into next, or start to develop things needed in each cell cycle, or in the phase that it's in. Regulation for proper functioning requires activation and deactivation and inhibition. So don't think of them as like more positive or negative than the other. It's just that sometimes, you know, like CDK, you might think that it's going to be a cyclin CDK, you think it's going to be active because it's got an activating phosphate on it. But if it also has the inactivating phosphate on it, then it's not going to be active. All right, well, thank you guys for coming. I'm going to stop the recording now.